All right, so this is lecture 4 from chapter 6. We're going to talk about a couple of key biochemistry techniques that are covered at the end of this chapter. You should be very familiar with these techniques because they're used frequently by biochemists. So some of these have already been mentioned within the chapter's text, but we're just going to talk a little bit about what each of these does, what the purpose of the technique is, and a little bit about how it's used. All right, so the first technique is circular dichroism. This uses the difference between the left and right-handed polarized light, which looks at the chiral environment that is produced by chromophores. And so in this case, the chromophore would be your protein. And if you remember, when we talked about amino acids, each amino acid is chiral. It either has a D or an L form, or RS, whichever convention you use. And that means that they will rotate polarized light in a different way depending on if you have the D or the L form of the amino acid. Now for proteins, almost all of your proteins are only going to have one chiral form of those amino acids that are present. But the rotation of this light is also going to be affected by the type of secondary structure that the protein is in as well. So the signals that we collect using this technique is going to be in the UV range. So we're going to typically look at 190 to 250 nanometer light waves. And we will measure the absorption at those measurements. And the CD signals from these peptide bonds are going to depend completely on the type of secondary structure conformation that the amino acids are in. So we can't use this technique to say anything about the tertiary structure. We don't know how those secondary structures elements are in relationship to each other, but you can use this technique to tell if your protein is folded and to be able to identify the percentage of the secondary structure elements that are present in your given protein. So this technique is often used in protein folding studies because if you start with your fully folded protein, you can identify how much secondary structure is present of each type. And then if you start to unfold your protein, so for example, you were adding a chemical reagent or you were adding heat, then you could watch the loss of that secondary structure element by scanning with a CD through using circular dichroism. So here's an example spectra. This is broken down into protein samples of different types of secondary structure elements. So this black one is random coil. That's a protein that doesn't have any secondary structure at all. The green is alpha helices. So if you have an alpha helix, you have a maximum absorbance at 190. And then this trough with a little bit of a peak here at around 212, 215. And then you can kind of see this is kind of a characteristic for an alpha helix. And then for a beta conformation, this peak shifts a little bit, so it's closer to like 195. It's not quite as high as the alpha helix, and then you don't have this double trough here. So when you take a protein sample and get a CD spectra of it, very rarely do you only have alpha or beta sheet secondary structures, so this is kind of like the optimal. But what you end up with is a trace that's kind of an average between these. And then we've taken and made computer algorithms that can recognize that data and kind of identify what percentage of that data is coming from beta conformation, what percentage is coming from alpha helix, what percentage is coming from random coil. And so we can take that and follow and measure what the protein is looking like. So if you were doing an unfolding experiment and your protein was mostly alpha helical and beta conformation, this peak here is what you would be looking at, and that peak would start to disappear the more and more unfolded and the more and more random coil it becomes. Another technique that's widely used in biochemistry is UV-vis light absorbance. We've already talked about this within the actual main lecture, but both DNA and proteins absorb light in the UV range as well. So for DNA, this absorbance is at about 260. For proteins, that maximum absorbance is around 280. So if you look at this peak, what are you going to notice? Well, you notice that for proteins, we also have absorbance from 200 to 220. That is the range that you're looking at with CD. So these techniques are somewhat related. You're just looking at different elements of the protein. So the absorbance from 280 is coming from tyrosine, phenylalanine, and threonine, or no, sorry, tyrosine, phenylalanine, and tryptophan. In DNA, those nucleotides also absorb, but they're absorbing at a different wavelength, so they're absorbing at 260 versus 280. For both proteins and nucleic acids, this technique is widely used to identify the concentration of the sample. Now, it only works if you have a very pure sample of your protein or your DNA, because any other protein or DNA in the sample is also going to absorb within those same wavelengths. So if you run an SDS-page gel, and we'll talk about that in a minute too, and you have a single band so that you can pretty much say, okay, most of my sample is this one particular protein, this is a very useful technique in order to determine its protein concentration. So you would use the Beer-Lambert law, which says that the absorbance of the sample is equal to this epsilon, which is the absorptivity coefficient. This is different for every protein, and we can look it up, times the length of the cuvette. So this is typically 1 centimeter, and then the concentration. And so you can solve for C if you know the path length and the absorptivity coefficient and have determined your absorbance of your sample. Another technique that's widely used is fluorescence. So fluorescence is a characteristic that particular proteins have. This is not a characteristic shared by every single protein, but some proteins do have a fluorescence emission to them. The most well-known fluorescent protein is green fluorescent protein. This protein was first isolated from a jellyfish, and it's really unique because the protein itself has a chromophore that causes fluorescence that is composed of amino acids that have been modified. So this protein is used throughout biochemistry and cell biology to investigate different protein levels. You can use it as a tag because the gene for GFP encodes a protein that fluoresces. So GFP was the first one identified. That is green fluorescent protein, and so it fluoresces green. However, there are a number of different fluorescent proteins that have either been identified from other species of jellyfish or sea life that fluoresce at different wavelengths and have different colors, or have been modified. The chromophore has been modified to make it fluorescent at a different wavelength. So you can see that now we have a wide range of different colored fluorescent markers, ranging from blue all the way through most of the colors in the spectra. The gene for GFP is really cool because it encodes the entire fluorescent protein. It's frequently used as a fusion protein, so you can take your protein of interest and then add GFP to the end of it. And so then when that protein is expressed, it's expressed as itself with GFP connected onto it. And the GFP still is fluorescent. And so wherever that protein goes within a cell, you can tell because that portion of the cell will light up green. So this is often used in studies to monitor protein localization in cells. If you look at your cell biology textbooks, those images with really nicely colored, brightly colored fluorescent images, they all use this sort of tagging technology. So what is fluorescence exactly? Well, fluorescence uses the absorbance of light, and so when that light is absorbed and excited, then the chromophore will absorb that light and then release a certain amount of it as visible energy. So you lose a little bit as heat, and then you release a wavelength of light that's slightly higher or lower in energy than the light that excited it to begin with. And so if you take your sample of a fluorescent protein and take an absorbance scan, you would see that it absorbs light in a specific range. In this case, this is GFP. It absorbs light at around 490. And then if you take that same sample and excite it at 490 and then measure the light that it is emitting, the emitted light is at around 520, and so that's this green peak right here. So each fluorescent protein has its own unique characteristic absorbance peak and emission peak. And so the emission is the color of light that you are seeing here. So the emitted light has this particular color. The absorbed light is slightly lower wavelength or higher in energy in order to emit this. So GFP, you can do really cool stuff with it. You can tag different proteins in solution and excite their tags separately. So for example, if you wanted to do a co-localization study, you could tag a protein with a green fluorescent protein and excite it and see the green show up. And then you could tag another protein with something that's red, and you would excite that with a completely different wavelength, and you can see where they are combined and such. So the last technique that this chapter discusses is SDS-page electrophoresis. We've already talked about gel electrophoresis and how that technique works. With SDS, the main difference, this is the typical technique used to look at proteins in a gel, and the main difference is the addition of this detergent in order to separate out your samples from each other. A unique thing about this is that a lot of times we add a compound called BME or another compound that reduces disulfide bonds. And so when you're running SDS-page electrophoresis, SDS will actually interfere with the bonds in the protein itself, so your protein is denatured into a single strand. And so when you run it on this gel, you'll run a molecular weight ladder, and then you run your sample, and your sample should run at the molecular weight that it is when it's linear. If you have disulfide bonds in your protein, then you also have to add a reagent in there to denature the disulfide bonds, or the bonds will stay together. And so in this example, you have this antibody that is connected, these two different chains are connected by disulfide bonds. So when you add a reducing agent like BME into the solution, you break these disulfide bonds apart. And so without BME, this antibody runs at 150 kilodaltons because it's the entire chain together. However, if you use a denaturing agent to break those disulfide bonds apart, now we have two blue chains and two red chains, and this thing runs at around 53 and 23. And to make the entire molecule, you would have two of these and two of these, and so you split them apart and it shows up as two bands on this gel. Now, an important consideration when you look at SDS-page gels is you really have to know a bit about your sample to know what you're seeing and understand it. So if your sample is something like this antibody, just because you have two bands doesn't mean your protein isn't pure. It's just a reflection of the fact that your protein chains are linked together by these disulfide bonds. So this is a commonly used technique in order to tell the purity of your protein sample that you're investigating, and incredibly useful because you can use it to tell the molecular weight of the protein that you're working with.